A lot of things can go wrong. For example, a history might not even be taken, so a patient can present, um, diagnosed, and treated and sent back without a history being ever taken. Another thing that can happen is that the patient can have traveled frequently between endemic regions and have forgotten which regions they have traveled to. Or another problem is if the location of presentation and the travel destination are both endemic, you can't really tell if parasites from where. Lastly, patients may have traveled for illicit purposes and so might be unwilling to disclose if they have even traveled. So with this, analyzing parasite DNA can offer a solution or at least facilitate the importation detection of imported malaria cases. By using parasite DNA, we can differentiate the parasites of each country. However, the GMS with regards to importation is unique in a couple of ways. So at the first level, we have different categories of importation. So we've already discussed and seen with a lot of malaria control programs here that the malaria here, the imported ones actually occur on the border. So these are cross-border malaria cases. And so importation is short-ranged, in other words. So these occur when, for example, migrant workers, mobile workers, they cross the border to do work, like, for example, plantations, or they go into the forests, you go foraging, things like that. And at the other level of importation, we have longer ranged importation. So, for example, people traveling between Southeast Asia and Africa, for example. The other point that makes the GMS unique concerning importation is that the region was actually the center of the emergence and spread of large international clonal clusters, specifically those that were resistant to dihydroartemisinin and piperacin. And this caused the genetic similarity of the region of parasites across the region to be very high. And so it makes it very difficult, especially with lower resolution barcoding biallelic SNPs, to differentiate between a parasite, between different parasites of each country. So to make things more concrete, how can we use genetic surveillance to help determine importation? So it's going to be useful here to talk about how we define relatedness, or how is it quantified. So given two samples and two DNA sequences of those samples, a high relatedness essentially means that the DNA sequences are similar across different loci. However, a low similarity or distant relatedness is defined when there's a high number of mismatches between the two DNA sequences. And because geographical distance and relatedness are correlated, we can see that parasites that are sampled, for example, in Cambodia and in Thailand, will share many common genetic variants compared to one that is sampled in Cambodia and in India, for example. And so using these common genetic variants, you can sort of extract a genetic signature of a parasite that is associated with a country, and that can tell you where the parasite came from. Now, what are the minimum requirements to reveal the association between a genetic signature of a country and that country, the origin of infection? So what you would need is a data set of sufficient size that contains genotyping data as well as geographical data. We would use this data set to train a statistical model, which is basically an algorithm here that learns the data and then makes prediction of new data, and which can tell you the country or region of infection. And we have that. We have the GRC, or genetic report card, which is a spreadsheet that the GenRe-Mekong team sends to its partners, and which contains geographical metadata and 101 barcoding genotypes for each collected sample, or in the case of PV, 99 microhaplotypes. So we can use this data set to train AI models and then use these AI models to predict the country or region of infection. And when it comes to AI, there are some useful definitions to learn about. So that will appear later in the slides as well. So a target variable is a variable that we want to predict, for in this case, the country or region. And a predictor variable is what the AI uses to make predictions, and in this type, genotype information. A classifier is a type of model that makes classification of countries or regions in this case. So putting everything together, we have two data sets. The PF-GRC data set, which contains almost 30,000 samples with 101 barcoding SNP genotypes and coming from 33 countries over 8 regions. We have the PV data set, which contains over 4,000 samples with 99 microhaplotypes coming from 4 countries in the Eastern Southeast Asia region. And then we're using these two data sets to train 3 different types of machine learning models or AI models. These are the random forest models, gradient boosting machine, and neural network. You don't need to know the details of how these work, but just know that they are different algorithms for achieving the same thing, which is to learn the input data and make predictions when data is inputted. So the objectives here is to develop a predictive model to predict region and country of origin of a given genotype. So we're using the 101 barcoding SNP genotypes to predict the region and country of PF samples, and the 99 microhaplotypes to predict the country of origin of PV samples. And before I go into the results, so I'll spend just briefly telling you about the process of how this is done. So we took data from the Malaria Gen dataset, and we used our own GenRe-Mekong dataset. Then we filtered to keep only high quality samples and high quality genotypes. And then we created 3 data sets for each research question. And for each data set, we trained 3 models each, and then we tested and evaluated each model based on data that we withheld in the training of the models. So in that way, it's not biased. So looking first at the prediction of countries with PF parasites. So out of the models that we trained, the best performing model was the gradient boosting machine, which achieved an 85% accuracy. Accuracy within East Southeast Asia will range between 84 and 89%, while for non-East Southeast Asia, it was 61 to 93%. Compared to other models, the gradient boosting machine actually performed the best within the East Southeast Asian region. However, it's still not at 100%. And so we wanted to look closer at what exactly is this model struggling with. So what are the misclassifications? And looking closer, we see that most of the mistakes this model makes is between countries in the Southeast Asian region. So, and especially between countries with known parasite flows between each other. So like with Cambodia, Laos, and Vietnam is one compartment, and Thailand and Myanmar is another compartment. So for example, this is a matrix that tells you how many of the samples the models correctly and incorrectly identified. So looking at Cambodia here, the model correctly predicted 381 Cambodian samples to be from Cambodia. However, it predicted 13 Cambodian samples to be from Laos, and 5 from Thailand, and 25 from Vietnam. But the overwhelming majority of the samples were correctly identified, and for all of the East Southeast Asian countries. So that's pretty good. So between Vietnam, Cambodia, and Laos, an average of about 12% of samples were incorrectly identified to be from the same compartment, but not the correct country. And as for the other compartment, Thailand and Myanmar, the average misclassification rate within the compartment was at 6%. And because in the training data set, we actually excluded samples that belong to internationally circulating clusters, this also sort of indicates the parasite genetic similarity between these countries as well. Now, next we wanted to look at the same thing, but for PV. So given a PV microhaplotype, which country is this PV case from? And so of all the models, the best performing model was the neural network, which achieved a 92.4% accuracy. And actually, all models performed quite well at above 90%. And similar to the PF, the models tended to misclassify samples with known parasite flows between each other. In this case, it was Cambodia and Laos. So here you can see 13 Cambodian samples were misattributed to Laos, and the same for Laos samples that were misattributed as Cambodian. So the increase in accuracy probably came from the fact that the microhaplotypes offered a much higher resolution for the models to learn from. So it was able to extract a high resolution genetic signature for each country. And for the last research question, assigning regions to PF samples, and actually the best performing model was performed at 100% success rate between differentiating between African and East Southeast Asian parasites. And this is great. This tells us that if a travel history is not taken, or if somebody went to Africa and lied to the doctors that they did, we could tell them that, okay, this parasite is from Africa. You can see the samples, East Southeast Asian samples, 98% were correctly predicted, but 0% were misattributed as being from all regions of Africa, Central, East, and Western Africa. No samples. So that's excellent. On average, though, across the regions, the gradient boosting machine, which is the best model, achieved a global accuracy of 89.4%. And for East Southeast Asia, the accuracy was at 98.1%, but it was slightly outperformed by another model, which performed better by only just 1%. So quite comparable. So how do we integrate this into NMCP workflows, or how do we make this interactive and accessible? So one of the things that we thought of is to develop a reporting tool that NMCPs can use to look at a sample of interest and generate a sample report such as this one, where it would tell you the metadata of the sample, so where it was collected, the date of collection, and the missingness of the genotypes. So this can tell you about how confident we should be in the predictions based on the genotypes. And it would also tell you about the prediction, the most probable prediction for region, country, and cluster, as well as give the prediction probabilities. And it could also tell you about the resistance profile of that sample for the different antimalarials. And if this sample belonged to a cluster, it could also show the epidemic curve of the cluster, so where this cluster has been and over time in the region. So we are actively developing this tool and trying to incorporate it into a web application, but it is still under development. So further work to be done. To sum everything up, even with 101 barcoding SNPs, which is considered a lower resolution genetic data, these models could still distinguish African parasites from Asian parasites at 100% accuracy. Our best models were the gradient boosting machine and the neural net, which all scored more than 85% accuracy, especially with the neural net, which scored an average of 92% in accuracy in predicting countries. And at this stage, using the barcoding SNPs to differentiate long-range importation from local cases could be the next step forward. So in future work, because of the limited resolution of the genetic barcodes, we're using whole genome sequences and microhaplotypes to train our models, and this will yield better results. Also, selection of new, more differentiating barcodes can increase accuracy as well. The goal here is to get country and prediction and region predictions at over 99% accuracy. Next, optimization of the PV models will yield more accurate results because actually the AI models of PV were actually not trained for as long as those of PF. And so that's just another low-hanging fruit that we can take. Just train the models for longer, they will yield better accuracies. And lastly, integrating our models into a reporting tool, for example, in a web application or a data dashboard, would be the next step and would make these statistical models more accessible to NMCPs. So that's all for my talk. I'd like to thank our sponsors and the NMCPs who are here, and lastly our funders, the Gates Foundation, the Global Fund, and the Wellcome Trust. Thank you very much.
Great, thank you, Ethan. Any questions for Ethan? Yeah. Yes, yeah.
Actually, it's not a question. It's because you use the available data set to train the model, and within that data set, the geographic data represented by the location of sample collection. Mm-hmm. It's not the place that the patient got infection. So how can you improve it with your model?
So you mean that the location on the data set is just the location where the patient presented and not the location of infection? So yeah, that's one of the assumptions going into this study. So we assume that wherever the patient presented, the patient probably got infected around that area. But actually, I also looked at potentially imported cases in our data set as well, looking at genetic distances. And I've noticed a few cases, not a lot, about less than 10 cases of Western GMS parasites in Eastern GMS. But of course, I can't tell if the Eastern GMS countries are imported between each other. So yeah, that is a limitation.
Yeah. Yeah. So generally, the assumption was that there's very few imported cases in the data set, which may or may not. Well, we hope that was correct.
Yes. Because most of the samples in the data set represented 2015 to 2020, where there's a high proportion of local cases anyways. We presume that the models will be trained on the real genetic signatures of that country.
But it's a very good point anyway. Yeah. Other questions? Anybody? Oh, yeah, another one.
And actually, because as you mentioned that it will be integrated for NMCP to exploring a case, then and the requirement is the genetic barcode.
Yeah.
So is there any option of a more simpler data set that they can input but still can find out that where the parasites come from?
You mean, is it possible to develop another assay to
No,
use?
because we require the genetic barcode,
Yeah.
but in other case they don't have the full 101 SNP barcode, but have a smaller set of the SNP.
Yeah.
Then can it generate to the high accurate prediction?
It would depend on the SNPs that those groups were using. If they're at a high enough genetic resolution to allow the models to learn the genetic signatures. But I would say like anything less than 20 genetic barcodes is not going to be enough.
Yeah, I think part of the future work here is to see whether we can find more targeted variants. But in fact, for the next phase, now that the number of cases is very low, it's also possible that we might go the other way and just say, well, since it's only half a dozen cases, let's do whole genome sequencing, and then we have more accurate. So it could go either way. Okay, any more? Right. So thank you very much, Ethan. And now for the next session, yes, sorry, John.
Do you know what the cost is for the different, if you're going to use a whole genome versus something else, or?
Yeah, I think Ian is better qualified than me. What's the difference in cost between doing genetic report cards versus whole genome sequencing? So can we throw a number out up? So it's something like $50 versus $100 or something like that.
Actually, with the current, based on current experience of what happened in our group, and because we run the amplicon sequencing to generate the genetic barcode with high throughput, so the cost is really acceptable. It's around 50 to 60 US dollar per sample. But for whole genome sequencing, if currently we still use MySeq, it's not very high throughput for whole genome sequencing, so the cost is quite higher, around 6 times comparing to amplicon sequencing. But in case of you can invest of the a more power Illumina, for example, a more power sequencing system with the we can run with high throughput like NovaSeq or upper level or a different kind of platform like OTN. I think that the cost will be reduced because the number we provide is just for research and in the optimization period, but for implementation and apply with high throughput, I think the cost will be decreased.
Thank you. Yeah, not all laboratory setup are the same. Depending on the type of machine, your costs change quite substantially. Yeah. Yeah.